KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Hi students, now in this video we are discussing anatomy of monocot root and dicot root. Okay, or anatomy of dicot root and monocot root. So both examples and explanations, internal structures I have given explanation in this video. So try to watch without skip. Thank you. Let us have a look on this. Students, now as I said before, so in this video we are going to discuss anatomy of dicotyledons and monocotyledons. So here onwards, so actually our class topic chapter name is anatomy of flowering plants now. So actually, so they have given a name, so based on this information. Okay, so now, so in this anatomy now, so we will be discussing about the dicots and monocots. So dicots know roots, stem and leaves, as well as in a monocots also root, stems and leaf we will discuss. So first of all, in this video we will be discussing which one dicot root as well as monocot root in complete video. So let us watch one by one. So first of all, for better understanding, first line for better understanding of tissue organization of roots, stems and leaves, it is convenient to study the transfer section of the mature zones. Mature zones means here for understanding point of view, that understanding also not for summa, so better understanding. Better understanding of tissue organization of roots, stems and leaves is it is convenient to study. So means if you would like to study about the better understanding of any particular tissues, so that are maybe roots, stems and leaf or whatever it is, so definitely, so we must feel convenience to study the which one transverse section. So transfer section means we will be calling no TSS section. TS section we will call no Adida. So of which one we should not take like unmatured one, we have to take matured zones. Now let us start to discuss about the anatomy of dicotyledonous roots. Anatomy of which one? Dicot roots, let us take first. Okay, now the TSS section or the transfer section of a sunflower root, let us take into our experiments or explanation. Why? Because so sunflower plant it is comes under dicot root, dicot plant only, no. So that's why sunflower root let us take for doing of or for studying of internal structures. Now let us start to discuss. Means here if you take root system, no. If you take root system, it consists of epidermis, cortex, endodermis, pericycle, vascular tissues and pith. Means here all these terms you may know before or you can learn now. Okay, if you know all these things before, okay, suppose sir, if you take internal structure of root, so definitely it consists of epidermis, cortex, endodermis, pericycle. Likewise, if you know the terms, so definitely it is easy to study. Suppose if you don't know also, now we will explain, no? So now you can learn easily. Let us start to discuss. So transfer section. TS you can see there. Transfer section is simply we will be taking as a TS of a sunflower root. So that gives so what is the information or what is the anatomical characteristics means the outermost layer, the outermost protecting layer. Here author they didn't give information regarding protecting layer, like that kind of information they didn't give, but here we can save in our mind epidermis, or epidermis we will be calling by one more name, epiblema. The outermost protecting layer is epidermis or epiblema. That will be already in previous videos we seen now. So here now what are the cells involved in the formation of epidermis? All are compactly arranged without any intercellular spaces. Likewise, while explanation of epidermal tissue system I have given explanation. So likewise, so here also epidermis is outermost protecting layer. So cells are elongated cells and compactly arranged cells are there without any intercellular spaces. And one neat bit also I said now, what is that? So here epidermal cells will not possess or not possess which one? Chloroplast. So chloroplast. So chloroplast and not present the chloroplast, but our chloroplast is present in few specialized cells which are present in the epidermis. We will be calling as stomata. Likewise, a few somewhat points we have learned now. So likewise here also up to here we can say. Next one, many of the cells of epiblema. So many cells, or what are the cells present in the epidermis? No. So protrude in the form of means so protrude means which will come out or which come out. So which will come out from the epidermal cells and form unicellular root hairs. This is the important point. Means here this particular unicellular root hairs which are coming from the epiblema, means origin of root hairs is there in epiblema or epidermis. Means here the root hairs which are coming from the epidermis root hairs and helps in different types of functions. So here unicellular root hairs means what are the root hairs which are coming from the epidermis? Definitely these how many cells are there? Only single cells. Uni means a single one, unicellular. Now here these are all my own points here. What are the points I have written now? So this root hairs origin is exogenous origin. Root hairs origin already. So these particular points we have learned in roots. So in roots or root of zone of maturation, zone of elongation, zone of meristematic there. So in regions of root of we have given explanation. So we learned a few points there. So those points I have repeated again here. So this root hairs origin is exogenous origin and helps in absorption of water and minerals from the soil. So means when these particular root hairs are present, or when these root hairs are originated from the epidermis, so such particular origin of root hairs we will be taking as exogenous origin and helps in absorption of water and minerals from the soil. So means when definitely these particular root hairs helps in absorption of water and minerals in the soil. So because of the presence of root hairs, absorption area definitely is increased. So means absorption area of the soil is definitely is increased. So thereby here what is the point? Root hairs or which are coming from the epidermis or which are originated from the epidermis. Next one origin is exogenous origin, and what is the main important points means why we are calling as exogenous origin means don't forget, suppose if the any kind of tissue systems or any kind of structures which are developed from the outermost protecting layers, so such kind or outermost layers, so such kind of origin we will be calling as exogenous origin, means the specialized structures which are coming from the outermost layers. So that's why that kind of origin we will be calling as exogenous origin. Now next one, what is the main important function of root hairs? Definitely helps in absorption of water and minerals. So and one more is because of the presence of root hairs, because of the presence of root hairs, definitely absorption area is increased. So thereby, so because of the presence of root hairs, a chance is there to absorb the more quantities of water and minerals. So that is a main important point. So in this occasion we are discussing. Next one, so second layer means just below the epidermis or just below the epiblema. So what is a layer we can find out means, so the cortex consists second layer which is present beneath of or below the epidermis. So this particular cortex consists of several layers of thin walled parenchymatous cells. So means here what are the cells are present in the cortex, no? So several layers are there. So if you see this particular structure, no, if you see this particular structure, so what is the information here we can be taken means, if you take this particular cortex, several layers are there, no? So diagram, so here you see that, so many layers are there. This one is a root hair we have been studied, and epidermis also we studied now only. Next one, second layer which is present just below the epidermis, we will be taking as cortex, no? So this particular cortex, several layered thin walled parenchymatous cells with intercellular spaces. Once you see there, so several layered cells with only thin walled parenchyma. So here the cell wall is thin walled parenchymatous cells with intercellular spaces. Once you see there between the cells, so we can find out the empty space. So what is the space which is present between the two cells? No, we will be considering, our subject point of view, we are considering as intercellular spaces. So thereby, so here in this particular cortex, no, definitely we can find out the presence of intercellular space. Next one, innermost layer of the cortex. From here onwards important. Lesson entire backbone starts here. Okay, so now innermost layer of cortex is referred as endodermis. So innermost layer, one more time, once you see here, this is cortex, no, this is innermost layer. White color is there, no, with the white chalk piece I have drawn. So this particular one we will be taking as endodermis. So this particular endodermis is innermost layer of cortex. Innermost layer of cortex is endodermis. Next one, it comprises a single layer of barrel shaped cells without intercellular spaces. One more time you see there, it comprises a single layered barrel shaped cells. Once you see endodermal cells, this is endodermal cells. Here if you see this, no, how many layers are there? Only single layered, only, no, only white color chalk piece color you see. So single layered barrel shaped cells, no, suppose barrel shaped, so we can find out like how it will be like some kind of vessels which they will store wine and all, no? If you watch different types of movies, no, they will be showing off so extraction of grape juice from the grapes, so they will be showing, no? So what are the containers are there, no? All those will be in barrel shape. Now here that you leave beside. Now endodermal cells or shape also will be barrel shaped cells. Now here without any intercellular spaces. My dear students, so here what is the main important point we have to save in our mind means cortex. If you see there, no, cortex cells, no, made up of parenchymatous cells and thin walled with intercellular spaces within. Intercellular spaces are present in the cortex within, but endodermis also is a part of cortex only, but in endodermis intercellular spaces are absent. Once you see there, without intercellular spaces means intercellular spaces absent only, no, that is only no meaning. So try to understand. So thereby in endodermis single layered barrel shaped cells are there without any intercellular spaces, means this endodermis is a part of cortex, but this endodermis is not possessing of intercellular spaces. Next one, tangential walls, parallel walls. Suppose if you take one cell, exactly at spotlight, once you see, So ఇది tangential, ఇది tangential wall, and పక్కతలి cells like vertical lines are there, no, these lines are radial. இப்போ tangential walls and radial walls of the endodermal cells have deposition of water impermeable and waxy material sovereign in the form of Casparian strips. So here main important underneath bit, what is the main bit we have to be discussed means, so endodermal cells, these endodermal cells means here only, this one only we are continue to learn. These endodermal cells are tangential walls as well as radial walls. If you see there, no, definitely deposition, so these are all having deposition of water impermeable waxy material known as suberin. So because of the presence of a suberin in the form of which one formation taking place, Casparian strips of formation takes place. So my dear students, what is the main important bit we have to be discussed here means, so these endodermal cells are made up of suberin, so in the form of Casparian strips. So here what we can be seen means the formation of Casparian strips we can see, and here these endodermal cells, so definitely water impermeable and waxy material, waxy material, so that is suberin, so definitely forms the Casparian strips. So here one important neat bit I would like to give, what is that point, particular point, no, இப்போ பாருங்க, once you see the diagram, இப்போ this one is endodermis, no, இப்போ இங்கே உள்ளே try to see inside, inside here protoxylem is there and here metaxylem is there. This metaxylem and here chinna xylem is there, no, that one we will be taking as protoxylem. Exactly to beside to the protoxylem here in a endodermis region, no, so we can see the specialized cells, we can see the specialized cells are known as passage cells. Specialized cells known as passage cells means definitely these passage cells which are present where exactly beside to the or nearby protoxylem, means here exactly at spotlight and here and here and here. What we can find out, passage cells, once you say passage cells which are present in the endodermis. Now entire endodermis is impermeable, but so these passage cells, what are the specialized cells or the parenchymatous cells are there exactly nearby the protoxylem. These are all normal cells, like normal cells, definitely involves in exchange or definitely involves in the movement of water and minerals. One time see here, this root hair is there. This particular root hair absorbs the water and minerals before we see, no. So what are the water and minerals present in the soil? So this all must move through, come like this, and suppose if this particular endodermis is impermeable, means what are the water and minerals coming from the root hair, how it will be enters into the xylem and phloem members, how it will be enters into the xylem, so we cannot. So at the time, no, here because of the presence of passage cells exactly nearby or beside to the protoxylem, so because of this passage cells, what are the water molecules and the minerals coming from the soil enters into the xylem members, because only passage cells. Why? Because these passage cells are like helps in movement or helps in the passage of water and minerals, so through the endodermis. So that particular point, neat examination point of view, important to try to say. Next one, next to the endodermis. So next to the endodermis means here what is the information we have to be taken, no, inside of endodermis. Next to the endodermis, a few layers of thick-walled parenchymatous cells referred to as pericycle, means within the or inside of the endodermis, few layered thick-walled parenchymatous cells we will be taking as pericycle. So this pericycle information also is important. So what is the importance of this pericycle means, once you see there, this pericycle helps in initiation of lateral roots. So initiation of lateral roots. Once let us see the information of or the diagrammatical view of a pericycle. So this is a pericycle. So here if you see clearly, here koncham pink colored layers are there, cells. So here one and only unicellular. Unicellular means uniseriate. Not unicellular, uniseriate. Uniseriate means suppose if the single cells arrangement in one serial order. Okay, so one row. So this particular like a group of tissue we will be taking as a pericycle, which is situated just below the endodermis. So now what is the use of this particular pericycle cells means helps in initiation of lateral roots. In 2017 NEET examination they have given this bit. Okay, pericycle definitely helps in initiation of lateral roots. So thereby here one point we have to discuss, that is so initiation of lateral roots is definitely is under the control of a pericycle, we see now. So thereby from here to lateral roots origination we can see. So thereby here the lateral roots origin is endogenous. So lateral roots origin is endogenous. So suppose once you see the root hairs origin is exogenous, and here suppose here any roots formation taking place from the pericycle, so such particular roots we will be taking as lateral roots. So these lateral roots origin is endogenous. Why endogenous? So these lateral roots are formed from the inner layers, or from the inner layers or inner walls. So that's why here the lateral roots origin is endogenous origin. And one more means not only in the formation of lateral roots but also involves in the formation of cambial or vascular cambial ring or vascular cambium during secondary growth. Means in a secondary growth also. In a secondary growth, so definitely pericycle involves in the formation of vascular cambium. Means so thereby here what is the main important point? Once you see, so it is xylem. So these are 4 members of xylem, and here this one orange color like a small cap like structures are there, it is phloem. So between the xylem and phloem, our knowledge point of view, suppose if it is open means there cambium must be there, but here in young stages or in normal roots means before secondary growth, if you see there we could not find the presence of cambium, but same cambium or the vascular cambium we can see during secondary growth at the time So this particular vascular cambium presence we can see between the both xylem and phloem. So that particular information, how it will be, the structure, the location, everything, so in secondary growth very exclusively explain. But now in this occasion, so what is the point you have to, in this situation, what is the point you have to remember? Pericycle definitely involves in the initiation of lateral roots, and one more is vascular cambium. So thereby, so vascular cambium and one more is lateral roots definitely formed from the pericycle. Next one, pith is pith exactly at spotlight chinna space we are finding small gap. So that particular gap only our subject point of view we are calling as pith. Pith or medulla, 2 names are there. Pith or medulla, it is a small, so that's why it's a small, or sometimes inconspicuous, means we could not find out the presence of a pith there. Okay, so means here this particular point also important. So pith is a small and inconspicuous, means will not be visible very clearly. Next one, the parenchymatous cells which lie between the xylem and phloem is known as conjunctive tissue. So means here once you see there the tissues or the parenchymatous cells which are present between the xylem and phloem. Once you see idi phloem, idi xylem. So here koncham gap I left gap is there. So here in this particular gap we can find out the presence of conjunctive tissue. Suppose here if I draw particular cells means this particular diagram will not be very clear. That's why so I didn't mention the which one conjunctive tissue, but now we are learning the presence of conjunctive tissue we can see between the xylem and phloem. Ipo idi xylem amma and idi phloem. No, here gap we are finding. No, so in between this particular gap, no, we can find out the parenchymatous cells presence. So these parenchymatous cells only our subject point of view we are calling as conjunctive tissue. So thereby now once you say, so conjunctive tissue which is situated between both xylem and phloem. So next one, there are usually 2 to 4 layers or 2 to 4 xylem and phloem patches are present. There are how many are there? 2 or 4 layers or the 4 xylem and phloem patches are present. So means once you see, so based on this information, so what is the point here we can say, no, in anatomy of dicot root 2 to 4 xylem and phloem patches are there means, so here what is the condition we can take? Tetra means so 4 only, no, tetrarch condition. Xylem condition we will be taking as a tetrarch. Tetra means so 4 only, no, so thereby we will be taking as a tetrarch. And later a cambial ring develops, so between the xylem and phloem. So later, not now, so during a secondary growth, now only we see now in this side. So thereby what is the point? During secondary growth, cambial ring formation or the cambial ring develops between the xylem and phloem members. So that one means the formation of a cambium only takes place during the process of secondary growth between the xylem and phloem members. Next one, all tissues on the inside of the endodermis means, so here this one is endodermis. No, next one, what are the tissues which are present in the endodermis? Present inside of the endodermis, all tissues together we will be considering as a steel. Steel definitely consists of a pericycle, vascular bundles and a pith. Definitely in our examinations I will ask this bit. So what are the tissues which are present inside of the endodermis? Inside of endodermis, so we will be taking as a steel, STELE, steel. So the combination of a pericycle, ipo endodermis ulla pericycle is there, no. Next one, vascular bundles are there. So the xylem members and phloem members together only, no, we are calling as vascular bundles. Aparam ulle pith also is there, pith or medulla now we see, no. So all together, so this entire information simply we will be calling as steel. So thereby here, so if you see that diagrammatical explanation, so here the root hairs are present or which are all or these root hairs are originated from the epidermis, and these root hairs helps in absorption of water and minerals, and origin is exogenous origin. Next one, epidermis outermost protecting layer, no, definitely helps in or gives protection. That's where that point you no need to discuss, we know very well. Next one, just below the epidermis, parenchymatous cells are there with the intercellular spaces. We will be taking as cortex, but innermost layer of cortex only we are calling as endodermis, but there is no intercellular spaces, and cell's shape is barrel shaped. This particular point also I will ask. So endodermal cells or barrel shaped cells, and impermeable to the water because of the presence of a waxy layer suberin. So next one, within the endodermis, uniseriate cells are present. That particular cells arrangement known as a pericycle helps in lateral roots formation as well as vascular cambium ring formation during secondary growth. Next one, xylem and phloem members present alternate radial arrangement. We see in previous video, radial in roots, no, definitely arrangement of vascular bundles will be like a radial apdena. So xylem, phloem arrangement, no, alternate arrangement of vascular bundles we will find out, no. So this particular alternate arrangement of vascular bundles, our subject point of view we will be calling as, or we will be taking as radial type or radial arrangement. So once you see there, metaxylem which is situated towards the pith and protoxylem which is situated towards the pericycle, you know. So this kind of arrangement of xylem members we will be taking as exarch condition. In the previous videos we learned no exarch condition protoxylem which is situated towards the pericycle and metaxylem definitely which is present towards the pitta. So this particular condition we will be taking as exarch condition. So this particular entire information already in previous videos so we have discussed. Next one, let us start to discuss about the diagrammatical information of anatomy of monocot root. So in a dicot root point of view, one important point one illa rendu important points. What are those? No, pith is small or inconspicuous, and the xylem members arrangement means here how many members of xylem members are there? 4 are there, no? So that's why here in a dicot root xylem members are 4 are there. That's why we will be calling as a tetrarch condition. Tetrarch. Okay, tetrarch condition. No, these two are main important points where in a dicot root. Next one, in a monocot root point of view. So in a monocot root point of view, so before start to discuss about the structural or the anatomical information, once let us concentrate on the information, so thereby so you learn the information, right? So monocot root information point of view, once you see the anatomy of monocot root is similar to the dicot root. So thereby when this particular monocot root anatomy is similar to the dicot root, no, so till now what is the information we have seen in epidermis, cortex, endodermis and pericycle and pitta, everything is common. Just where once you see continue to read that one, similar to the dicot root in many aspects, means in many aspects means maximum information. So between the between the dicot root and monocot root will be same only, but a few exceptions are there. So in terms of pith and in terms of number of xylem members only difference, so remaining ellame same da. So that particular information only let us see. So like how dicot root consists of all these structures, means what are those? Epidermis, cortex, endodermis, pericycle, vascular tissues and pith. No, likewise monocot root also consists of all these layers, and then these two lines are backbones. Now as compared to the dicot root, so as compared to the dicot root, so which have fewer number of or fewer xylem bundles, fewer only, no, very less, just 4 only are there. So that's why here we are using the word fewer, means kammi, fewer xylem bundles, and they are usually, but here if you see here in monocot root, no, but they are more than 6 are there, 1 2 3 4 5 6 likewise. So here the 6 are there, so more than 6 suppose if we are finding, means so such kind of xylem bundles we will be calling as polyarch condition. Polyarch. Suppose a tetrarch means TETRA. tetrarch, இது common da. Polyarch means more than 6 are there, means இது polyarch. இங்கே 4 மட்டும்தான் இருக்கு, that's why here tetrarch condition. Okay, next one. So here the polyarch condition we are observing in monocot root. Next one, pith is large, ரொம்ப பெருசா இருக்கு, but here the pith is very small. So that's why here in monocot root, no, pith is large and well developed. இது neat bitama. Okay, so pith is large and well developed only in case of monocot root. Okay, monocot root. So thereby, so here in first line, what is the point we have discussed? Monocot root anatomy if you see, or dicot root anatomy if you see there, no, there most of the points are similar, but 2 important differences only are there. Once you concentrate on the diagram now, so here 4 xylem members are there, that's why xylem arrangement is tetrarch condition we are calling. But in a monocot root, no, here more than 6 are there or 6 are there, no, so such particular thing or such particular vascular bundles arrangement we will be calling as polyarch condition. Next one here, in a dicot root, no, pith is very small, but in monocot root, no, pith is large, pith is there and well developed pith is present. So well developed word author was used only for monocot root pith. only for monocot root. So thereby here, my dear students, so 2 points try to save more and more. What are the points? No, here the xylem arrangements is or the xylem numbers are tetrarchic number, whereas here polyarchic number, and here in a dicot root pith is a very small or inconspicuous. Pith is very small and inconspicuous, but in a monocot root pith is large and well developed. So these are all main important points related to the dicot root and monocot root. Suppose here, suppose we are not having public examinations, but suppose the same kind of, or suppose if they ask dicot root, suppose if we have studied about the information of dicot root, suppose if you don't know information of monocot root means, so we can write down the entire information of dicot root, but while writing xylem and pith matum we have to concentrate. So in a monocot root, pith is more than 6 xylem bundles are there. No, that's why polyarchic condition we will see. So that one we will differentiate, and pith is very large and well developed. This is only these two only main important differences, and remaining all points are similar to the each other. So this is a small explanation of both dicot root and monocot root. Try to watch this video. So while watching, no, take the base of or take the help of diagrams, so thereby you will understand very clearly. So suppose if you have any doubts, no, so no worry. So when you will come to the school at the time, suppose if you need explanation one more time also, we are ready to explain. Thank you and be safe at home.